Hello, and welcome everyone to this video podcast for human genetics. This will be our first podcast for the course where we're actually going to start talking about content that is relevant to human genetics. In this podcast, I will introduce many terms and concepts that we will come back to throughout the course, but this will provide a nice background for what we will learn throughout the semester. I want to go ahead and take a step back and begin by taking this rather larger picture. And as you can see here, I'm going to start with the planet Earth. We know that as we look at this Earth, that human populations are found throughout most of the land masses found on Earth. However, it's important for us to recognize that humans aren't the only occupants of this planet. In fact, we really make up a very small portion of all living organisms on this planet. Moreover, humans have been on this Earth for a relatively short period of time throughout Earth's history. However, this being a human genetics course, we're going to focus primarily on humans. So let's begin to narrow down here, and let's take a look at this smaller population of humans. Now, when we look at each of these individuals, we see many similarities between them. However, we also see many different characteristics. There are differences between the sexes of the individuals. The individuals all have different hues to their skin pigmentations. They have variable amounts of hair. Some have lots of hair and some have relatively small amounts of hair. We can see some variation amongst the heights of the individuals and the weights amongst the individuals. We can also see that some of them are wearing glasses and some aren't. They also have many other characteristics that are different that we just simply can't see by the simple observation of these individuals. Now, as we look at this population and we see these differences, we might make the assumption that at the genetic level, there's a lot of variation as well. However, what we now know is that amongst all of these individuals, there's probably only about a 0.5% difference at the genetic level. In fact, you could take any two individuals, say somebody from Mount Pleasant, Michigan, where I happen to be at, and compare that genome, human genome, to an individual that happens to live in Bombay, India. And you'll still see only about a 0.5% difference. And the reason for that is that humans are a relatively new species on Earth. We're a young species. This being a human genetics course, now I'd like to spend some time and focus on what it is that accounts for these similarities and these differences between these individuals. You've probably all seen a picture similar to this, and what this is a human karyotype. Each of these represents chromosome pairs of the karyotype. And I could circle all of them, but I'm not going to. We call all of these autosomes. Let me go ahead and try to write that here. So chromosomes 1 through 22 down here are all autosomes. Now the chromosomes here, the X chromosomes, and if there were Y chromosomes in this individual, they would be here. It just so happens that this is a female, or came from a female, and so therefore we don't see any Y chromosomes. But these are called sex chromosomes. For right now, what I'd like you to know is that autosomes make up the first 22 pairs of chromosomes in humans, and these other chromosomes, the X and Y, are called the sex chromosomes. The Y chromosome, as we'll see later in the semester, is primarily responsible for determining the sex of the individual. A genetic counselor might use a karyotype like this to determine the relative health of an individual. That is, are there any noticeable chromosomal abnormalities? Now, for us, we might look at this and it might be hard to tell, but you might think maybe the X chromosome is a little shorter here. That might be significant to the health of this individual. However, it would take a trained individual to really point out some of the finer details of this karyotype. I'd like to erase what we just had and now point out something else here. As I mentioned before, these chromosomes represent a pair. They're the exact same on both sides. And we'll talk in a moment how each of these chromosomes are made up of genes, and the order of the genes, say on chromosome 2, is the same on the left side as the chromosome 2 genes on the right side. The only difference initially is that one of these, say, came from the father, and the other came from the mother. And we'll talk about this in a few podcasts from now, about how chromosomes are divided as egg and sperm are made. But for now, recognize when you look at this karyotype, what you're looking at is half of the genome, half of the chromosomes that came from dad, and half of the chromosomes that came from mom. What I'd like to do now is to go to my whiteboard and break down these chromosomes and expand on what they actually are and why they are important. So what I'm going to do first here is just draw a typical person here. And one thing you'll recognize throughout the semester is that my artistic abilities are amazing. So don't be too jealous. What I'm going to do here is I'm going to take, say, one cell from this individual. So here's that cell. And within this cell is going to be the nucleus. And what I'm going to do is I'm going to take a chromosome out of this nucleus. Now remember, within this nucleus contains all of the chromosomes we just saw on that previous karyotype. In fact, we need to remember that of the trillions of cells found in this fabulously drawn human being here, they have the exact same number of chromosomes. But we're just looking at one of these. And I'm not going to pull out the whole karyotype, all the chromosomes, and draw them here. Rather, I'm just going to draw a single chromosome. And we can call it chromosome 1. It doesn't really matter for our purposes right now what it is, but let's just for fun give it a name. We'll say it's chromosome, and I'll abbreviate it just CHROM 1. Chromosomes have a few landmarks on them that are important to know. The first is at the end of each of them we have a telomere. We'll talk more about telomeres later, but for right now just know that telomeres are found at the ends of the chromosomes. You should also know that where the chromosome appears to pinch off, this is called the centromere. Often when somebody draws a cartoon of a chromosome, they put the centromere in the middle, but we'll learn as the semester goes on that every chromosome is characterized by the placement of that centromere. Some chromosomes have the centromere towards the end, some actually have it all the way towards the end, and some have it somewhere in between the end and the middle. But for right now, I would just like you to know that the centromere is located at the part of the chromosome where, as we draw it, it looks like it's pinched in a little bit. And we'll learn more when we talk about cell division and meiosis, why the centromere is important. Now, it's not always easy to see, but one of the key points that I think is really important to know is that it really is made up of one single but large double helical DNA molecule. I'll just draw it like so. It's not a whole bunch of DNA molecules, it's one large molecule. In fact, let's go ahead and write right here components of a chromosome. We're going to simplify here, but for now, let's just say you need to know that a chromosome is made up of one DNA molecule and a collection of proteins. These proteins play many roles. They may function in a structural role, maintaining the shape of the chromosome, maintaining the DNA. They may function to replicate the DNA during cell division, replication, or they may play a role in gene regulation. I won't draw the proteins here, but you should know that they're wrapped all the way around it. And throughout the semester, we'll come back to some of these important proteins that play roles in these three pathways. The next thing I'd like to talk about is the gene. We'll expand on what a gene is throughout the semester, but for right now, what I'd like you to know is that within this chromosome, there are many genes. And remember that this DNA molecule, I drew it rather large here, it's very compressed. So there are several genes within this chromosome. So let's write that here. Genes found on chromosomes. Genes make various products. We think one of the main products they make are proteins. But we also know that not all genes found on the chromosome make protein. Some just make various kinds of RNA. Some of those RNAs are important in making proteins, but other of the RNAs have different functions. And we'll talk about some of those functions throughout the semester. In humans, we know we have between 20 and 25,000 genes. Now, not every chromosome has the same number of genes. For instance, chromosome 1 has the most genes. It has approximately 2,000 genes. Chromosome 18 and 21, they each have approximately 200 to 250 genes. The X chromosome has approximately 800 genes. The Y chromosome is the smallest of them all, and it only has 60 genes. It's almost an embarrassment to call it a chromosome. It's so small. But it has some important genes on it that play a significant role in determining the sex of an individual. While I said before that any two individuals, their genetic makeup is very similar, it's also important to note that there are differences. So in our little cartoon up here, let's just say this is gene 1 and this is gene 2. And let's just make person 1 and person 2 here. While they both have a copy of gene 1 and a copy of gene 2, there may be some differences between gene 1 in person 1 and gene 1 in person 2. Now we'll talk about the components of genes in a little bit, but for now I'd like you to know that they're made up of things we call nucleotides. And we'll talk about nucleotides in just a moment in more detail. But they're either adenines, cytosines, thymines, or guanines. And so let's say gene 1 of person 1 looks like this. And gene 1 of person 2 looks similar, except that these two bases are different. And that's because in person 2, this gene was mutated. So that's called a mutation. Anytime the nucleotides change, it's called a mutation. Now there are other ways, later in the semester we'll break down mutation in different ways, but for now, let's just recognize that a change in nucleotides is a mutation. Now, from a very broad perspective, sometimes a mutation, even if it's just 1 or 2 bases here, can lead to disease. It can also lead to a better function. And if that better function allows that organism to reproduce more, then ultimately, within a population, that can lead to evolution. More often than not, mutations within a person's DNA has small or no. As I said, sometimes these changes can lead to diseases. We know that a single base pair change from one person to the other can lead to diseases like sickle cell anemia and other diseases. Okay, I went ahead and erased gene 2 here because I just really want to focus on gene 1 here, because I want to introduce a new term for you. And this term is allele. An allele is an alternate form of a gene. So if this by chance up here was the gene for, say, cystic fibrosis, so I'll write CF for cystic fibrosis under the name gene 1 here. And let's pretend in this example that this is the sequence that you would see in the gene, which would be much, much larger than what we see here. But this is the sequence that you would see in a healthy individual. And then let's say that this mutation, this change in nucleotides, results in the disease form of the cystic fibrosis gene. Now these are both still cystic fibrosis genes, but they are different alleles. They're alternate forms of the same gene. We're going to use this term here, allele, quite frequently throughout the semester. Okay, there are 2 more terms I would like to introduce you to, as we'll refer to them frequently throughout the semester. The first one here is genotype. And the next one is phenotype. The genotype are the allele combinations in an individual. So allele combinations in an individual that causes a trait. Don't mistake this term with genome. Genome includes all of the genetic material. Genotype is just allele combination. So, for instance, this person here in gene 1, they would have 2 of these. That would be the genotype. Just that allele combination in that one individual. So phenotype is the trait expressed from the genotype. In a general sense, you can think of the genotype as the DNA portion of that gene, and the phenotype is the product of that gene, which is often the protein. Now, as I've gone through this introduction here up to this point, I realize there's a few terms that I did not fully define for you that I think are important for you to know as we proceed with this course. The first term that I want to make sure you know the definition of is genome. We've hinted around the definition of this, but I haven't explicitly said what it is. A genome is a collection of all the genetic information in an organism. This includes all the chromosomes that you saw on that previous image of a karyotype. However, there are other sources of DNA in an organism that we should place in the category of a genome. For instance, within this cell here, we have mitochondria. And mitochondria have their own DNA as well. Therefore, it's part of a human's genome. Now, interestingly, this genome contains protein coding portions, but also non-protein coding portions. Still part of the genome, though. Interestingly, at least I find it interesting, is that this protein coding portion in a human genome is only 1.5% of the genome. The other 98.5% make up this non-protein coding portion. It's still very important, this non-protein coding portion of our genome. It just doesn't make a protein. It's important in regulating genes and other functions, some of which we know and some of which we're still a little unclear. However, one of my pet peeves is this: never call it junk DNA. That will make me sad. Let's put a big cross through that. Junk DNA, that term implies that there's some parts of the genome that we don't need. And we don't know if that's true. In fact, most often we find that the entire genome is important. So next, I'd like to define the exome. It's similar to the genome, except that the exome only looks at the 1.5% or so sequence that encodes proteins. Let's call this the collection of protein encoding DNA. So if we were to draw a long piece of DNA here, it would have several protein encoding segments on it, which we would call genes, as I show here in red. So the exome would only be these sequences here, whereas the genome would include this whole region. The part of the DNA that has the protein coding portions, the exome that is, and the non-exome parts, the non-coding portion of the genome. Again, the protein encoding portion of this genome, the exome, is only about 1.5%. Okay, there is one last thing I wanted to mention before we end this podcast, and that is something known as the central dogma. I'll refer to this at various points throughout the semester, but this is one of the central themes to genetics. And so it's important to have a base understanding of it, and then we will, of course, build upon this as we move forward in this semester. In its simplest form, the central dogma is going from DNA, and we can think of the DNA as the gene or genotype that is then transcribed to RNA, and then that RNA is then translated into protein. And you can think of this protein as the phenotype. As the semester moves forward, we'll spend a lot more time talking about this central dogma. Okay, well, now that ends this podcast. As you're moving forward, if you have any questions at all, please don't hesitate to email me or ask members of the class, particularly those who are in your team. Okay, well, that's it for this podcast. Bye for now.